Good morning, friends of the Falkor. Coming to you live for 2,000 meters below the seafloor, or below the sea level, not below the seafloor, in the Gulf of California. So this is the good ship Falkor, and a good ship she is indeed. And the ROV SuBastian is down at just over 2,100 meters. We are in the Gulf of California investigating hydrothermal fields in the Pescadero Basin, a small little pull-apart basin where new oceanic crust and volcanism is occurring, but underneath the sediment cover, we've been investigating hydrothermal vents here. We are headed back to the Auka field, the first one discovered here in 2015, and as I've said before, it was discovered on April 12th, 2015, which was my birthday. We are going to land a few tens of meters, 75 meters away from Diane's vent, which was visited earlier this cruise. We're going to be trying to collect some Xenophyophores, very strange little single-celled animals, colonial, or I guess they're colonial. Strange looking little creatures that look like little sponges on the seafloor. Not sponges as in the animal sponge, but more like just a dirty, porous little mat. So hopefully we will see some and core them and bring them back for study. And then we will go try and take water samples in Diane's vent, which is a very vigorous, it was 289 degrees when last measured in 2018. We haven't tried to sample or measure that yet, so will be toward the start of the dive. And then we'll move over to an area of the Auka field that we haven't seen yet on this cruise, an area that we're calling Dolomite because the mineral dolomite was seen in a few samples taken from there. So we'll be investigating that. It's an area of low mounds and tube worms and clams, and so looking forward to seeing that again. This is my fourth dive cruise, or cruise with dives to the Auka field. So I was here in 2015 with the Western Flyer, the Monterey Bay Aquarium Research Institute ship. I was here 2017 on the Nautilus with the Woods Hole group. We came here on the Falkor in 2018, and now we're coming back in 2021. So we've got sort of a 6-year record, and based on what we've seen so far, a lot of the things like Diane's vent look exactly the same as they were when we last saw them or first saw them, and other places have changed dramatically. So these systems are quite dynamic. Sometimes they sit at steady state, and other times they change. So that's what we have in store today. Got J-Rod flying the sub, taking beautiful pictures of a creature. You know who that is? Is that some kind of little shrimp?
Amphipod?
Amphipod, maybe. And that looks like an anemone.
Doing like a hobo crab thing, or an hobo to
yeah,
like a fence?
I don't know what that is. Why those seem like two separate creatures, but they look like they're connected.
The crabs on the bottom that hold on to an anemone on their back?
Yeah. I've never seen that done by an amphipod, but that certainly seems, maybe he's just taking it home for dinner for the family.
Yeah.
So we've got about 1,500 meters to go on the bottom. I am not going to bore you with a bunch of nonsense that I know nothing about. We'll just let you enjoy the biology, but if I see anything that I think I can comment on, we'll jump in the mic. Other than that, just enjoy the show. That's weird. I've never seen that before. So here we have a small creature with 4 little lobes on it. Must be a Xenophore.
Start swimming. Make the rainbows.
And it is not true that I work for mining companies because I'm retired. I don't work. I just come out here and have fun.
Yeah, I thought y'all like the gray chair better. Sorry? I thought y'all like that other chair better. Platypus. Not that 8 years? I thought they were the only one. Keep it. Knuckles from the Sonic Hedgehog. Okay. There. Whatever. Float is this one. Or mural.
So that little polychaete seems to be buoyantly hanging there. It must be a floata and not a sinker. want that?
Yeah.
You want a second one?
Yes, please.
Do that.
Same cast, that's fine.
We are on the air.
Oh,
If you would like.
comment. So this is a floater. I can't give you a species name. It was first described from Chile, and it's never been collected from down there.
Stone jar one.
What would you like to call this one, Greg?
Floater 2, please. Thanks, John. And we've sequenced one from California, and
Oh man,
Was it?
I went the wrong direction.
Yeah, was it?
Yeah, I went the wrong direction. Put it in 5. Oh,
Yeah. That's all good.
wow, it's big.
sorry, Greg. I put it in the wrong jar.
Oh, we'll pay attention.
Which jar is it in?
It's in jar 5.
5.
Jar 5. Gotcha. That's PHLOAT.
FLOTA.
Sorry, Greg.
And it's in a different group to the swimmer worms, and we're on a lookout for a swimmer as well. And I'd like to solve the problem of the floater name, but until we get the Chilean one, we can't know if the California one or if this will be the same as the California one. So I'm looking forward to getting that up tonight. We've got 2 now. And on our way down, we'll keep an eye out for the swimmer worms. Again, we've only sequenced and collected those off California. Several from the Western Pacific, if you were along for our Northwestern Australia Indian Ocean trip last year. We got 4 new species on that trip, thanks to the skills of J-Rod and his team. And I've got a student working up those new species for his masters. Yeah. We've got about 1,000 meters to go, and this is the zone where we might see the swimmers, but we're getting a bit deeper. It would be interesting to see how the floaters are going to have to go down to 3,700 meters and then all the way back up. So I don't have high hopes for how they'll take that journey. These creatures, swimmer and floater, from their 2 respective worm families, have benthic relatives, and in both cases they evolved from benthic ancestors, because we've done a pretty detailed evolutionary tree of each group now. And in the case of swimmer, if we get a good look at one, they have 8 little bombs along the side of their heads, which are actually gills, and they can shed those and they glow bright green. And when we look at their relatives, they also have those 8 gills, and they also fall off very easily, but they don't glow, and they're usually long and filamentous. And if you want to look up a creature called the squid worm, that kind of shows you an intermediate form in a way between the benthic forms and the pelagic swimmers.
Cool. Those are related to the swimmer or the floater?
To the swimmer.
Weird,
So
'cause
squid.
it has the really big flippy things
Yeah.
like the floater does.
Yeah.
Big flag.
Yeah. Yeah, but they're completely independent. Swimmer and squidworm go together. And floater goes with a whole bunch of weird sort of sediment-covered animals, but they have this gelatinous matrix that, of course, you don't see floater having it covered in sediment, but on the seafloor, when we catch these benthic relatives, they're all sandy. And then there's another one called Poebius, Poebius, and that, for a while, no one even realized it was a worm related to floater and swimmer. And it's because it doesn't look segmented, it's just kind of this floating blob but it has tentacles and gills, and eventually people realized that it was another relative of floater, but not the same group as floater. They both came independently from the benthic. So this colonization of the pelagic ecosystem by annelid worms has happened probably about 10 to 12 times.
Is there like a major characteristic that
That's
separates them?
the Poebius worms look just like they don't have all those segments and swimming bristles that the swimmer and floater have. Here we're zooming on a house of a larvation, and this is sinking slowly to the seafloor, and it's a good source of marine snow, so Victoria wants some of these. Jar 2 for this?
Yes.
Oh, it really fell apart. Yeah, well, it'll end up in the bottom, and they'll be able to...
Is this marine snow one?
Yeah,
Yeah.
S2, slope 2.
In jar 2.
Yep.
What time are we supposed to go test? Now?
Oh, yeah.
So I think,
Yep, we're now.
You guys got
Yeah, that first photo was shallower than I think we've seen it before. Not positive.
Yeah, it was about 2,000.
about to run out of here.
Breakfast hot dogs are my favorite thing. I didn't even know you were allowed to do that.
Very good. Look in our number 1.
So there's a floater in jar 1 already.
Yeah, they
Okay.
said that everything can go in the same.
Cool.
Number 1? Yeah. Let me get a better shot of it.
Yeah. I'm not sure what it will look like, but there's a ways to go.
Get ready to turn the Catholics back on now. Right before I go. Just about to focus. Go. No. I'll let you know, like right before I go to San.
So,
That's him, right?
that's a swimmer. Yeah.
All right, Catholics on.
Well, it's our lucky morning today. This can go in 1.
Yeah.
Same.
Yeah,
Okay.
I'm in there right now.
Great.
A little bit of the
To guess how big it is, I'd say it's only 3 centimeters long, based on the nozzle size. They get a bit bigger up north.
All around. Stop the picture.
A lot of the ones we see are near the bottom, and they probably go down on the bottom and feed on the sediment and then swim back up. But this one's way too high to be able to do that, so it's probably living off marine snow.
It is.
Good job. Thank you.
He had to be chased down, didn't he?
Yeah.
All right, he's in.
Well,
Next
we'll
thing-
hopefully that'll make it to the surface. We'll
all right.
sequence it and figure out if it's a new species or one we know from California.
All right, that's me on
Yeah,
D.
the floaters were very big, 15 centimeters. $2,700 meters, pretty deep for the swimmer too.
Do you want another one if we see another one?
Sure. Sometimes in California we'd see several different ones at different depths.
If we find another one, do you want it to go in jar number 5 with another photo?
Oh, that could be a good idea. Yeah. But don't hold up the descent. It doesn't.
No.
It doesn't interfere too much, right?
Okay. No,
You
I stopped the one to the one.
but we've got plenty more days to get more. They have separate sexes, so it's nice to get a male and a female. So yesterday we ended up with a male Xenophyophores, and I was able to really look at that compared to the female that we got the day before. So that was
The
really,
female was the day before?
yeah,
The one that was upside down?
yeah. J-Rod picked up a couple of swimmers back in July, August. We were off California, and I thought that would be one we knew, but it turned out to be a new one. So I don't know how many species of swimmers you've collected now. It must be close to 4. 3 off Western Australia.
The orange bomber one? Is that
The
his
orange bomber, yeah. That one was a known one. That's the Philippine bomber. But some of the ones you got from Bremer, the southern coast, and one of the ones from northwest was a new species. And there's a new species of swimmer, squidworm as well. That was new.
That's awesome. Just so many little things.
Yeah. I've got a student at Scripps, Paul Proctor, and he's doing his master's on the swimmer worms from Australia. Yeah, we have the swimmers from Australia there. If you go back, I can't remember the dive, but it was when they were in the Bremer Canyon. So that would have been early February 20 or January 20.
Yeah.
And they came across thousands of bombers just above the seafloor. It was an amazing sight.
Yeah, they were everywhere.
That's one of the new species. Yeah.
Oh, that one was.
Yeah, that's a new one.
What about this guy?
That's a swimmer. Or a little floater.
Like a little.
Yeah. No, we can pass on that one.
Okay.
It's two's great. Wow, yesterday you saw nothing, and today?
Yeah. So crazy.
So we're at 2,900 meters. We've still got about 800 meters to go. We're heading to a vent today called Diane's Vent, or the vicinity of Diane's Vent. We've got a few targets down there. Looking at microbial mats, putting out some experiments, picking up some rocks. It won't be as visually as spectacular as yesterday's amazing edifices, but still should be an interesting dive as well. We're all looking forward to lots more samples today. But Victoria will be in shortly, and she'll give a more detailed overview of the plan for today.
Second of April.
Oh, it was April.
That's what Simon says.
Yeah. Okay, I'll look that up.
Is when we did the other bombers?
April 2020. No, I'd left the ship and come home for COVID, running away from COVID by then.
Yeah, I got off in
And there was one right before that as well.
What's this one?
Well, that looks like a swimmer. Yes. So much marine snow in this place. That's why we can support these kind of creatures. Yeah, that can go in 5. That'd be great.
Right with that. You get a better shot of it?
Oh, yes, please. Its head is down. You can see it's got little feeding appendages. Little bombs, you can just get a glimpse of the little green spheres on each side. That's great. Look at that. Mostly they'll drop off on the journey, but hopefully I'll get a couple.
Catherine's back on. Alright, this is 5, 6, 5, 6, 4. How's it? Oh, come on.
Hmm.
Fancy flying, Jeremy.
Oh, yeah. It's hard to imagine how difficult this is to do.
Thought he was going in the hole.
Oof, just touched it.
Oh, yeah.
Fantastic.
Nice flying.
And it's in the canister. And it's actually in the canister with a floater.
Nice. Greg, are you just calling all these S1?
Um,
What do you
I'll
want?
talk to you in a minute about it. Oh, that's great. Okay, we're now at 2950 meters, and it's a question about the water temperature. It's 1.8 Celsius. But we'll be seeing some warmer water today. Not super hot today, probably. So there was a breakage.
That's a nice way of saying I messed up. When I put the legends on my
Is it really 473? Okay, so our dive numbers are off. So the dive number is 474, but it on our overlay says 473.
Is that right?
Or are we at 473? What's our dive number?
474. Yep, sorry, I didn't change that on there.
Okay, thanks. Will that affect anything so far?
No.
Okay. Seems like the
Which one? It just
the sample sea log.
On my overlay.
It just updated.
What about sea log?
Yes, 474.
And was it taking 474 prior to that change?
Yeah,
It was?
That's just my overlay.
Oh, got it.
That I forgot to change over.
Okay, no problem. I see. Sea log is similar to that last one was a swimmer? That's amazing. What a collection. Swimmer with one M. So I've never heard you pronounce. I always called them Phoebius. Oh, Phoebius. Yeah, they're very cool. They were the first pelagic polyp I ever saw.
But we've got one. Not our chapters. It says it's published, but we got one on Chile.
I didn't know that it was related, yeah. Didn't see.
Two different branches out of it.
Okay. Oh yeah. So section sample 2, the depth was 2612, and it's green. It's a larvation, like a yellow wheat larvation. Is there anything else in there, or is just that one? You have the depths on yours. The one they got first. It says, it doesn't say either, it says just section sample 5 is the first sample. Section sample 5.
There was a splitter before that in section 2.
Okay, that one,
Correct.
and that one,
Oh, sure.
and canister, spin around up top. There's a little thing right there, behind the
Oh,
top.
nice. Yeah, here you can see, is that what the audience is seeing? The swimmer's being shy, it's behind the canister intake, but you can see the contrast in the body size. And there's a nice big floater. Wow. There's stuff on the bottom of some tubes that came through the hose in the morning.
from last night.
from yesterday's dive.
Yeah.
We're just feeding them.
Yeah. Just giving a little bed of straw to sit on. It's going to be a long day for them.
And there's some up top.
Yeah, and there's the swimmer. So there's a probably 10 times as big, the floater over the swimmer.
And toasting that thing.
What's that?
And toasting that thing.
Studs? Yeah.
I should
Figure out if like I didn't put all the way over. But it didn't. But I don't know if it'll just go to space.
I'm not going all the way over, it's fine.
You do it hurts already. Told her like it's a call. Okay. I know, but yeah. I still hurt. You're happy sitting here for a bit. That's mine, right? That's your mind. Got it.
Did you do an overview? Zeke, did you do an overview?
I did, but not many people were on.
Do you want to do another one and talk about Diane's vent?
You
Go ahead. You better stop sampling and get to the point.
Okay, so we're about 500 meters or so from the bottom. And our plan, for those who joined in late, we're coming down into the Auka Vent Field. And we're going to land about 75 meters away from a very high temperature vent. And we're going to be looking for small little creatures called Xenophyophores on the way in, try and sample a couple of those. And then we're going to sample Diane's vent, which is a very vigorous high temperature vent. Was 289 degrees when last measured. And we'll do a temperature and try and collect 2 water samples there. And then we will transit over to sort of the middle part of the Auka Vent Field, moving over to the east and look at an area that we're informally calling Dolomite because 2 samples collected on previous dives showed Dolomite, which is a magnesium calcium carbonate at that site, whereas almost everything essentially everything is precipitated directly from the hydrothermal fluids is calcite, just calcium carbonate. And so the biologists and microbiologists want to know what's different over at that site, so we'll be investigating that. And before we go over there, actually there's a very robust, fairly high temperature bacterial mat just to the south of Diane's vent. We'll stop and do several push cores in there for microbiological studies and measure the temperatures while we're coring. So that's the plan for today's dive. Everything gets done early. We may go over and take a good look at the P mound, which we haven't seen yet on this cruise. It's north of and somewhat similar to the Z mound, which we visited several days ago, and it will be the site of some biological sampling later on a later dive. So that's kind of our plan for today. And the way we're going to try and sample the Xenophyophores is to just core over them, and then bring them up on the top of a sediment core on top of a push core. in the overlay you see the background map in color is the 1 meter gridded bathymetry that was collected by AUV here from the Falkor in 2018. And then the darker overlay is actually a photo mosaic, which was also collected on that cruise. That was a very novel mapping technique where we had a tool sled on the bottom of the ROV SuBastian and flew at about 3 to 5 meters above the bottom, and it had combined multi-beam sonar, laser LiDAR, and stereo photo cameras. And so we were able to make a map that can be gridded at centimeter scale. I think we have a 5 centimeter gridded version of that bathymetry, and we can lay the photo mosaic on top. There's the 5 centimeter scale bathymetry. So you can see, compared to the meter scale, we're getting much more detail. We can get centimeter or even sub-centimeter vertical resolution out of that survey. So you can actually see clam tracks in the bathymetry. That's how fine scale that bathymetry is. And then photo mosaic allows us to see what the images of the seafloor look like. And the brighter areas you see are typically where there's bacterial mat or tube worms, which are sort of light, and the darker areas are mostly sediment covered. So we have a highly unusual data source here in terms of the level of detail of mapping. This has rarely been done anywhere on this kind of scale. The mapping was done by Dave Caress, who is the chief scientist for all 3 of the legs here. Dave is back at MBARI, Monterey Bay Aquarium Research Institute. He was out here on the ship on leg 2, but is back watching and directing dives from his office in MBARI. He plans to come back with the MBARI ship, the Western Flyer, and their submersible dock rickets with a newer version of the deep high resolution mapping system. When we did this, the mapping tool was basically fixed, looking straight down at the seafloor, right on the front of the sub. The new mapping system will actually articulate. So that means it can swing up and down. And so, for example, as we climb up something like the Z mound, which sticks up, the mapping sled will articulate and move up so that it's imaging ahead of the sub instead of straight down. So it follows the topography and will give us a much more accurate view of the seafloor. So the work that Dave and his colleagues at MBARI are doing is truly phenomenal in terms of providing levels of detail that really just don't exist in very many places. And the beauty of that is that we can now start to do biological and microbiological experiments and have them in context of the larger geology and biology of the vent field. So rather than looking at little postage stamps from pictures, we'll be able to see the bigger picture. So it's been really fun to try and to be part of this team, be able to come out with all these really talented people and learn from them. And it's one of the things that really enjoy coming out with a multidisciplinary team, is that everyone has different expertise, and you always learn things from your colleagues when you come out, and that's one of the really fun things about being here. And I assume that the reason you folks are watching this is because you enjoy learning that kind of stuff too. So we're certainly glad to have you here. There's a nice 3D rendering now with the low mound in the foreground is, in fact, I think, yeah, that's where the Dolomite sample, I think, comes from. So that'll be where we're heading later in the dive here.
1 meter, 5 centimeters.
Okay, so I'm toggling back and forth between the 1 meter bathymetry
Okay, Rich.
and the 5 centimeter bathymetry. So
Hey Jared, I just got to bring the heading around slightly forward as the wind is starting to pick up a little bit.
Right. How much you thinking?
Probably 10 to 15 degrees at least.
Okay, roger that. That should work out just fine.
Okay, thanks.
and so in the more vivid colors, you're seeing the 5 centimeter resolution, and the big mound there is the Z mound, and you can see all kinds of talus blocks that are well defined on that. Out in the distance, you see the P mound straight north of the Z mound and over to the left of the C mound, and you can see those are 1 meter resolution. So you can see they're similar size and shape, but you look at the difference in the detail. Here's the 1 meter bathymetry over the Z mound, and now look at the detail you can see with the new 5 centimeter resolution. So these are really fantastic maps to be able to work with.
It's a fun 30.
So our depth of the landing site is something pretty close to 3650 meters, so less than 200 meters off bottom now.
My hand back up. Put my ear back up for you.
So we've already sampled a floater and a swimmer. I think here we have a wiggler. Do you know who that is?
I have no idea.
Yeah. None of the biologists are in the control room right now, and none of the rest of us know exactly what that is. It certainly is worm-like, and it's got little appendages, and other than that, you know as much as I do. Actually, probably know more than I do.
All right, I'll go
Did you see that?
Yeah, so it was a polychaete.
Okay.
Probably came up from the seafloor.
Oh.
What's the depth there? 3660-ish.
Yeah, 3650, so something give or take a few.
So.
We won't count. We won't. Exactly.
So, right here, where I had that little marker, you're starting there. You're going to be working on the seafloor, looking for Xenophyophores platforms, going to Diane's vent. Diane's vent, water samples, then going over to the Dolomites for rocks. Pilot. Heading is changing, our boat is changing its heading a little bit more, so keep an eye on that. And jars. 1, 2, and 5 all have really, 1 to 5 are things that can swim back out, so we just leave it on 8 at all times until we need to go. That makes me feel better. Cool.
So we've just done a little cruise ship. Adam is now in the pilot seat, and Chris is over on the minutes. And John is running all the high-tech video and things, and I'm just sitting here blathering on as usual.
Doing a fine job.
Sitting over there with the best of
Yeah. Keeping us into it.
I stopped. It was stopped. I just couldn't stop.
So the bottom water temperature is about 2 degrees. That's what we expect in these kind of deep waters. Surface water is very warm. It's in the high 20s, maybe almost 30 degrees C. So the big challenge is we'll try to bring up things like the swimmers that we have captured. They handle the pressure change pretty well, but they don't like the temperature change. So we hope to get them up in cold water quick as you can, getting through the thermocline so that they are in good shape when they come up.
You do whatever you need to do, buddy.
You do what you need to do what you need to get.
Okay, we're starting to get an altimeter hit, so that means we're approaching bottom. Should be something like 60 meters off, which is just about the depth we expect to be landing in. It's always nice when things agree with what you think they ought to be. And if we've done this right, we'll come down in a nice flat sediment covered bottom, and there'll be a Xenophyophore right in front of us when we land. And given that I'm a geologist and geochemist and don't describe new species, I will not be naming new species after anyone or anything. That's the job for the biologist. My expertise, if I have any, is understanding water chemistry and rock chemistry and mineralogy and how they go together. So I'll be in charge of trying to get a sample of the hydrothermal vents. Actually, that's a lie. The pilots are the ones who are going to be in charge of getting the sample, but then they let me have it, and we'll take it back to the lab. I'm working with several other people on this project, including Jeff Wheat, who's an adjunct at Embari and is a professor at University of Alaska at Fairbanks, but lives down in the Monterey Bay Aquarium area. He'll be doing some of the major elements of chemistry on the water samples, and Marv Lilly, University of Washington, will be doing the gases, and tomorrow Baron Baumberger, sorry I mispronounced the name, and Noah will be doing helium isotopes on this. So rarely does one person do anything in isolation in science these days. It's always a team effort, and I'm just pleased to be part of the team. All of the vent material, most of the vent material is calcite. That is the more stable polymorph of calcium carbonate. Another form of calcium carbonate is called aragonite. Some creatures make their shells out of calcite and some out of aragonite. But the vents, being high temperature, gives things time to come to equilibrium, and the form of calcium carbonate we see in the vents is indeed calcite. And some of them form beautiful crystals, beautiful rhombohedral crystals that are just as clear as a nice diamond. They're really very pretty samples when they come up from the seafloor. There's the seafloor. It's all flat, sediment covered. Very little in the way of creatures right here. This is Galathea, just went off the lower left, lower right corner of the screen. And our chief scientist, Victoria Orphan, is here, and I'm going to turn it over to her, and she's going to find a Xenophyophore for us.
Tall order. No, no.
We're going to start off with some white balancing here, make sure that the colors on our video are giving us the best quality possible and more accurate representation of what we're really seeing down here. Morning, everyone. All right, so keep your eyes peeled. We're looking for basically little mud balls on the seafloor that are single-celled protists, large, multinucleate, that are called Xenophyophores. And these are foraminifera that are agglutinating, so they build their tests or their house out of sediments. And they have really amazing, beautiful, intricate structures that help, we think, with water flow so they can collect more particles. And we've been studying these in a variety of deep sea environments and looking at the microbial communities that are living in these houses with the animal, and trying to understand how this might help with their cycling of nitrogen and other compounds. Is that one there? Stop. So. Stop.
Trying to decide if it was
I think it
Xeno
is.
or
I think it is.
Thorian excrement.
So it's just off to the lower left corner. Yep, that is
Yeah.
one. All right. Behold, the magnificent Xenophyophore. Two of them. Yeah. Okay.
What's amazing about Dave Caress's maps is that we can target.
There they are. So, what may look like a blob of sediment is actually a giant protus. And they're pretty fragile creatures on the seafloor, on the sediments like this. The ones that live on the rocky hillsides of seamounts are a little bit more robust. You can pick them up with the manipulator arm, but I think with these we're going to try to push core over the top of one. So, a push core, maybe one of the polycarbonate ones. Can we put the lasers on too so we can get a size?
Not number 7.
Yeah, not 7.
Not 7.
No. Thank you. Is that gonna fit in the push core? Oh, how about the one in front of it? Let's see.
That's that for sure. I mean, it'll fit. It's not so good.
Yeah.
Oh, you'll get some of it.
How about this
That
look?
one
This little cutie.
maybe.
They're so weird here.
I know they're a little bit more
They're
fragile.
They're
Yeah,
muddy.
they're really muddy compared to the sea mermaids.
Yeah.
I can't seem to. Yeah.
Gosh,
Alright, did you get a photo?
tricky
Yep.
business.
No, they want off the ground plastic. Yeah.
Man, the only way is push core, I know.
Yeah.
Yeah, that's not. You're not going to pick that up successfully.
Gosh,
It's just.
so odd.
It's just mud.
Yeah. A web, mud web.
Okay. Alright, should we give it a shot?
Yeah.
Alright.
It's going to be interesting to try to incubate those.
Yeah.
Alright.
To the core. A lot of space.
Yeah.
But you don't care. You don't care about much more than like
That's
5
right.
centimeters of mud, right?
Yep. Yeah, so maybe like half sediment, half water, so we have a nice big interface to work with. Yeah.
Yeah, so the push core ID is 7.5 centimeters.
Yeah.
So anything
Yeah.
bigger than.
All you need is a little plug.
Yeah.
Plug in the bottom.
You know, we could try to pick the other one up with the manip and put it in a quiver.
Yeah.
We might as well
I
try.
think it's worth a try.
Since
See
we're
how-
parked.
It's useful to see how it behaves too. I remember being surprised
They stuck together.
when it was possible.
Yeah.
Really?
Yeah.
Yep.
Roxy was definitely can do that. That was the preferred method.
Yeah, no, and I'm even some we picked up in Monterey Canyon. You can even do it with the manipulator, but these look
Yeah, these are going to be
pretty
tricky.
interesting.
Like the size of
The other cool thing about these Xenophyophores is that they, in turn, provide habitat for other organisms. So our colleagues at Scripps, the ecologists there, have shown that fish will lay their eggs in these structures. We often find brittle stars and other organisms that live within the matrix. So it's a whole little ecosystem in and of itself. And we've recently found out that they have a very specific group of microorganisms that are in association with it, and we don't fully understand the activities these organisms are doing, but we have some hints based on the genomic data. And so these samples will help us to test some of these earlier hypotheses about the interactions between the microorganisms, the bacteria and the archaea, and the microeukaryote, the protist itself. All right.
Wait a minute, something doesn't feel quite right. No, they're kind of
Beautiful.
jiggly and distinct
Yeah,
from the sediment.
very nice sample.
Push core sized. That's nice.
Yeah. How convenient.
All right. Half and half, right?
That's perfect. Yeah, thank you.
As long as it holds in. This is the problem that we had in 2018.
Really? Does it slip out? It's not muddy?
Yeah.
More sandy?
This is coming back to me now.
So it's coarser
I think
than
sometimes we find we have to like give it a shake in there
Yes.
to kind of break the suction, and then it'll stay in there.
That's right.
That is
That's
what
right.
Chris
Definitely
just did.
played
Yeah.
this game before, where
Uh-oh. Uh-oh.
some
Uh-oh. Oh.
yeah, so it is super fine stuff. I think you got to go. I know you want
Even
your interface,
deeper.
but I think you want to go deeper just to get
Yeah.
enough friction to hold it in there.
Yeah.
Yeah.
The push core.
No, I see it on the floor
Yeah,
there.
so you can get-
Just, we might be able to
Let's try
pick
scooping
them
the
up.
other one.
Could
or
we,
picking it
do you
up.
think we could pick it up with the clam scoop
Yeah.
and put it in a quiver?
Do you want to try again since you're here with the
Yeah,
push
we
core?
can, we could try again. Try to,
I think if you go a little bit deeper, you'll
Yeah.
be fine.
okay. We just need to make a
Yeah,
note
I
that,
thought when I was first started coming up with it, you could see it started to drop,
Yeah.
and that's why I went back in and
Yeah.
maneuver deal, and I thought that's
Make a note that it
Yeah.
might be upside down.
no, that the sediment isn't
Oh.
exactly, it's
Yeah.
it's okay though, it's close, it's probably
Right.
enough. Yeah. All right, take 2.
Whoops. Oh, in the meantime, Victoria, people want to know if you think that there is life under the ice moons of Europa and Titan and those.
Oh.
you have no landing on your roof.
That is a great question. That you can break this down into 2 just separate questions that are related. One is whether or not there are conditions that are habitable for life as we know it that might exist under the ices in the oceans of Europa, or perhaps on Enceladus. The other is whether or not you might find actual life there, and that's a much harder question to address. And so, again, looking at these places, these extreme environments on Earth, like these hydrothermal vents, gives us a way of constraining the potential zone of habitability for life as we know it. And of course, when we talk about the champions of extreme living, frequently these are the microbial organisms that are pervasive on our planet. And so I can't answer the question of whether or not there is necessarily going to be life under the ice caps of Europa, but it provides a lot of interesting.
Uh-oh. Slipping.
Oh,
There you go.
okay. Don't dawdle. Go.
It's a race
Shove it,
against
shove it,
time.
shove it. Go.
I don't know.
I don't know if I made it.
I don't think so.
Oh.
It's no.
I think we lost it.
Yeah. But that's all that's in the quiver now, so might as well just stow it.
Maybe it's in there. It came in and we're just gonna
And stayed in the middle.
Stayed perfectly in the middle. It's got a core over it.
Different tactic. Let's see.
All right.
Good try, Chris. Though.
Yep.
It was a race against time.
It was a
Valiant.
race. Okay, how about can we,
Oh, he's got to push.
oh yeah.
That.
J-Rod has fans. It's not J-Rod. Our pilots today are Chris and Adam. Currently.
Chris and Adam, yes. Yep, there are 5 pilots out here with us today, and we get the great pleasure of interacting closely with all of them throughout the day as they go through their rotations. It was mentioned earlier, we're sailing with a reduced number of personnel, and for COVID reasons, and so this has increased each of our workloads, and the pilots also are sailing with fewer than normal pilots, which means that we get to see them more frequently as they rotate in and out.
Yeah. Mud.
Yeah, that would make sense. There we go.
ROV Bridge.
Yeah, go ahead.
Yeah, and I'd just like to bring the heading around a little more to port so we have a northerly heading. Did you guys want to move my waypoint accordingly?
Yeah, that's perfect. Thanks. I'll move it right now. Thank you.
Alright, cheers.
Given how easily you found those, I would just hold that core tube and go find another one.
Yeah, the problem is it was so full of sediment, even if we got another one, we had to.
Yeah, but I think if, hold that core tube, some of that'll wash out, then we get another tube and just make
Yeah.
sure it'll be fine, I think.
Yeah.
Yeah.
Well, we're gonna try to use it. Well, they're putting it away now.
Yeah, that's, yeah,
Yeah.
but I don't think you're gonna wanna dig that thing out of there.
No, we're not going to. Just want
I'll
to
bet
not
we
lose
find,
the
I'll bet we
shore.
find some more.
Yeah.
Yeah.
There's another one here, so we'll try to see if we can scoop it.
Give it a scoop, that's what I want, yeah.
Since it seemed pretty
Yeah,
more robust
the
than I
way
think.
that one held together, I think was good.
That's very
You
promising.
want to put on the sample number, put there. Okay, yeah, that's fine. Go ahead, yeah, submit. Let me show you, and then if we see any more, go to invertebrate, and then down here. So you can just check that box. Yeah. When we see it, you can cancel that one. But
All right.
when you take photos,
Should we try a different tact?
You want to do a little scoop of this?
Little,
and try to put it
little
on the quiver?
scoopy. Yep.
great.
All right.
All right, maybe it's going to be the quivers.
So it's pretty amazing. So we had noted we saw Xenophyophores here in this general vicinity in 2018. So this is why we started our dive on the flats heading into Diane's vent, and we were able to find some more really quickly. What I didn't pay attention to was the notes that we took about the sediment being extra fine and falling out when we tried to push core, and so definitely highlighting that in our notes today, so we'll make sure we use the proper tools to collect Xenophyophores in the future.
Obviously, you responded. What do you? Is it still we stand by that name?
Yeah.
Okay. What?
Of this whole exchange.
Did you see that on the Slack? It's all good? Okay, we're sticking with it. It's good to get the feedback.
Oh yeah.
Who does those measurements at Caltech?
Antoine did them over material science.
Nice.
Yeah.
It's great to know.
Yeah, really that was for our internal, so we knew where we're going. It wasn't.
It's on the back plan.
Yeah.
Yeah, we can. Did you want to try the scoop, or did you want to try to like just get under the dirt and?
Well, Chris, what would you like to try? Yeah.
Yeah.
Use your pool noodles to grab it. All right. Hold your breath.
Not even the scoop. Oh, the finger pool noodles? Finger
Yeah.
noodles?
Yeah. Worms are like, we're out of here.
It's a big.
Well, the question is, what do the Xenophyophores eat? And we don't know this well, so the biology, the physiology of these organisms is still...
No, we
no,
don't want
we
the
don't.
dirt.
We like dirt.
Yeah, that's better.
I think it's best, and you could just like dump off the Xeno or without
Yeah.
the dirt into the canister.
Well, whatever works. Whatever works.
Yeah. Let's see. Might be intact. Might be there. It's still there. It's still there.
Still
Or
there.
Is it too? What do you think about dirt
I mean,
getting in amongst them?
It'll
That's
wash
that's
out.
fine.
Wash out. Okay.
Yeah.
Like, is it? What is the structure?
It's agglutinated sediment. So the organism, it
No.
is no, it's hard.
It's hard.
I mean, not super hard, Chris. But
So the ones that we collect out on the seamounts, you guys were able to actually grab them with the manipulator without
Yeah.
crushing it. I mean, I think it's worth trying because the worst thing that happens is that we lose it, and then
Well-
we've learned something.
but why is everybody against the clam scoop?
Oh,
Too much sediment ends up in the quiver.
well, if it works to grab it with the manipulator, that has been more successful for us even than the clam scoop.
What do you
If
got,
it's-
Mama Bear?
If
clam
it's
scoop?
too fragile for grabbing, then I would go with the clam scoop.
And I bet they could delicately use the scoop so you just don't get much sediment in it.
Yeah.
Plus you can rinse it out.
Yep.
Shake it out. Oh, look, we have a Gallifreid coming over to wonder
Taking
what's going
advantage.
on.
Oh.
Well.
Oh.
It's very soft.
Okay. Oh.
I pick it up.
How about half a? Oh.
Yeah, no, these are too soft.
It's okay.
That one didn't look rigid. It looked fudgy.
It's a
It
little.
Yeah, what are they talking about?
Yeah.
So we went from the biggest single cell animal to two of the biggest half cell animals.
What?
We went from one of those biggest single cell animals to two of the world's biggest half cell animals.
Okay. Sponges. So how do you collect sponges on the seafloor like this?
I collect the whole thing with some sediment. I'm gonna try before.
Yeah.
Yep.
You do the scoop and just kind of go like this and filter out the sediment.
That's what I was thinking, but
Yeah.
Can you do
um,
that?
But Chris is
But-
proposing
but
maybe
Chris-
like the ice cream scoop.
yeah, go for it.
I'll try like a
Try it.
time, just
We have faith, Chris. Nope. So I think the current thinking about how these guys feed is that they are filter feeding particles out of the water column. But again, very little is known about these organisms. They're actually considered to be an indicator species of these fragile deep ocean environments, and so we're interested in better understanding a little bit more about their ecology, and from our perspective, what kinds of microorganisms. Oy. What kind of microorganisms do we find in them? We may never know.
That's why I never played over here.
I scoop this.
All right. So it's actually good to have a little bit of challenges and failures here, because the pilots make everything look super easy, and it's anything but. And so you're seeing sort of the true side of the challenge of doing work in the deep sea with these manipulator arms here.
And we know that we've done some 16-inch bar coding with them, so we know the microbial community lives in them, but it's kind of unusual. They like concentrate certain microbes in their tissues.
Seamounts.
Seamount, yeah, I know. We definitely could get them on the seamounts.
That's easy.
And it would be great. I mean, we still would like to have sediment. I don't know if we want to take. We should try to take a full push course and just so we know if we can get it. If we can get it, then
Yeah.
I think Gina would be happy with that.
There's some nitrogen cycling
Okay.
microbes within them. Let's see.
All right. So we tried push core. We tried manipulator, and now we are going to go with my favorite tool, the clam scoop, or we'll call it the Xeno scoop.
Mama bear clam scoop.
Mama bear. This is the medium-sized clam scoop. There's a little.
Can we get the gallon to grab it? And we'll bring the gallon.
Yeah, we might get a twofer here.
Yeah, crab is looking for handouts.
Oh,
Nobody ever trusts me.
the pointy, but you know, it's a different kind of clam scoop. This pointy angular clam scoop, I'm really digging.
I know.
It's a great design.
We could have one
Made.
Made.
Yeah, for the doctor.
Bring it with us.
Yeah. Oh, it slid in the back.
Yeah.
Now you can knock that sediment off.
Just deploy the clam scoop at
Yeah.
Ziva to pick it up in April.
Oh, that'd be great.
Yeah.
It would be a colonization experiment, and we get the scoop back.
Randy before.
All right. Clam scoop for the win. Well, shouldn't say that. Let's get it in.
Yeah, whoa, Victoria.
I know,
Oh,
I know.
yeah,
That was
just-
bad. That was...
Did you knock that sediment off, Chris?
No.
No, it's fine. We'll take the sediment. Take the sediment. Just.
But wait, but maybe the Xeno is more buoyant, so
No.
it might dump.
It'll go in.
I just thought maybe he could like tennis toss it in over the sediment. No, that's not your. That's not in his training. Huh.
I'm sick to damn tennis tossing. I've never seen him play tennis, but
Okay,
Yeah.
what's the other?
Yeah, it's more.
Badminton.
It's more like paddle ball.
Badminton. Yeah. Okay. I was sick the day of tennis tossing.
Get on in there. Yay! All right.
Half a Xeno.
Very good.
Okay. Success. Okay, maybe can you fly with the clam scoop, or you need it to close the quiver? Yeah, to close. Okay.
I'll sit it down and pick it back
Okay.
up. I'll sit it and say use.
Yeah.
Hey. Yeah, it's an animal. We'll call it A1. Today.
So that's quiver 8, right?
I'm gonna go ahead and set up for another one that I don't mind taking a tap off
Yeah.
some time.
No problem.
7.
Yeah, that'd be great.
Remember to filter the corners.
Oh yeah, we're gonna try to target the corner ones so we can get the quivers out.
Yeah.
So 7 and 8, 1 and 2.
Yeah.
It fell over.
So you're gonna target the corner ones for.
Oh, just the
Ease of
four
access?
the four middle ones are hard to pull out.
Yeah, we actually, even J-Rod was trying to help, but we couldn't get number 4 out last night, so. With these guys, we need to pour them out. Reaching in.
Right, so 1-4.
Yes, please.
No, 474.
That's how many are enabled?
Yeah.
That's one.
Is that one?
That was great, folks. Maybe could be one just up. Yeah.
Yeah.
Z wants to make sure he can keep his bio badge. He's on the lookout for more Xenos. No. Where are you looking?
Almost directly in front of the gas station.
Oh, maybe.
the first two things. Okay.
I think that could be one. Right up ahead. There's a few there, yeah. Yep. Perfect. Yep, go for it. There's at least 3 there.
Yeah.
474. Gotta put a post-it.
All right. Seems like a good morning for Xenophyophore collection. Having better luck with Xenophyophores than I am with mushroom hunting. The morels of the sea.
Oh,
Yeah.
it's big.
Yep.
Yeah, no kidding.
Can you get a twofer? A double scoop?
Welcome at this point. Thank goodness for a current. Clear up the.
Yeah, every time I sit down in this mud, it all sticks to the vehicle.
At least it's
Yeah.
moving.
Yeah,
Though.
it's going on.
Yeah, it's current, nice current.
That's always the problem.
We're gonna wait
work.
for this to clear out a little bit. Another fun fact about the Xenophyophore is they have this most beautiful aroma. I'm not kidding. They actually smell like mushrooms,
Truffles.
truffles. They have this really earthy, beautiful smell, and they exude this kind of golden brownish liquid. You leave them in
Would you
ethanol
recommend them in
for
pasta?
a while. We haven't tried to cook with them, no. But,
Definitely not xenophile truffles. I think there's a sea pig right there.
Okay,
He's
thanks.
snuffling for truffles. Yeah, he's right in there.
Yep.
We'll see.
Yeah, Z is saying there's a sea pig that's hunting around, snuffling
Cucumber.
around for truffles.
Truffles, snorkeling, sea pig.
We do see pigs and xenophyophores in the same environment.
Oh, this could be
man,
a new
we've made
thing.
A new
There's
association.
one, he's in the cloud, he's right, he's
Right
in the
there?
right corner.
No. Maybe we can hire him.
Yeah.
Put a little leash on.
Yeah.
Truffle dogs of Eastern Europe.
Well, George, next time I'm up at Mbari, we'll bring a Xenophyophore and we can cook it up together. Yeah.
Gritty. I'm gonna go.
We hear
with
they
gritty.
good for fiber intake.
Come on, just take this one. Here, do you want me to keep hunting for another one? Or are they few and far between?
I don't know. Yeah.
Is this just gonna keep happening, though?
Yeah. If we land again, are we gonna get another pile
Game
or?
of-
If I could fly through the water and get some of this sediment off the vehicle, it'd be
Yeah.
fun.
Okay, we can try
I
to
don't
clear.
want to leave it if you think they're not going to find anymore.
Well, is it not clearing? Let's just.
It's clear up here, but it's
Yeah, it's still coming through.
Yeah.
Can you change any lighting? Okay, so I still can't see him. That was a little better, actually.
Yeah.
Oh, can you go to the dial cam, Chris? Let's.
Yes.
No, man, not got to see it yet.
Sit.
It's getting
Yeah.
better.
Keep-
Can's worse. I've invested this much time. Let's see. Oh yeah, I think we're gonna. It'll be okay.
It's
Let's just stay. Oh, yeah. Okay. I see him.
yeah. I see him. Can't you just feel your way?
Yeah, yeah. Don't you have feedback on the manipulator? Tell?
Just have
You know, bulldog?
Yes, please.
Seems good.
Wayfarer.
Yep.
ISO down.
Does that help?
Should I take a picture of this?
Hello, Brett. Our pilots are indeed amazing.
Send it to Marco.
Yeah. The pointing tip I love.
Yeah. Half the battle is finding the right tool for the job, and of course
Going
having.
deeper though, might just
Yeah,
be breaking up if I don't.
Yeah, that's okay. Maybe we can kind of slowly jiggle it, try to get some of the loose sediment out.
You're gonna get both in there. Yeah. Yeah, there's two. Eyes are bigger than your stomach, Victoria.
Shh. It'd be like panning for Xeno gold, just shaking out or sieving flour.
Not when you could knock off without all that stuff.
Xenophyophores are not protected or endangered as far as we know, but they are considered an indicator species for fragile habitats in the benthos. And so they are one of these organisms that we want to learn a little bit more about their ecology. Might help us with preserving these ecosystems and developing policies that try to minimize potential impacts that humans may have on the seafloor.
They open 7 again. 7's already put in some. No. We haven't put anything in it.
The first A1
Went in for 8.
and then it opened 7. This is the first.
Are you trying to find a Xeno toss?
Again,
See it? Over the top?
Yeah, exactly.
Yeah. That's in.
Oh,
Oh.
3 more pieces of it.
Oh, there was one. Sorry, I was thinking
okay.
one down there.
That's okay. Maybe one will fall. Nope.
They are lighter.
All right. They are. So just for giggles, let's see if we can get the one.
Hold on, okay.
Right in front of us. Yeah, it's not. It's not.
Hmm?
Oh, it's there.
Right there.
Yeah.
Oh, you're right.
It's just left of the trigger on the gas side.
Yeah, it's right here.
Yes, I think. That's what we're looking at, yeah. I think there's it. Maybe not like it in the mud.
Yeah, I don't think that was.
That's not
it.
it.
It was to the right of that. It's just that the porch, when they pulled the drawer out, it covered it.
Ah.
That's what I
Do you want to push forward gently?
Yeah. Or we can keep.
It might still be there, but
Do you want to bump up and just see if you can find a?
You think it's still there, Shauna?
I don't know. I thought it was to the right,
Or did
and
we
I
cover it?
wonder if you pulled out the drawer, if it covered it a little bit. I actually think it was more to the right of us.
Hmm.
Because he was scooping over here and kind of.
Yeah, I can't see. Yeah, why don't let's bump. I mean,
There were more. You've seen
there's
more.
there's more.
Yeah.
Hopefully.
It's there in a little bit now.
I'll just fly gentle. We'll leave that quiver open.
Yeah.
For the next one.
Yeah. Could be one there.
The autofocus. Maybe
Yeah.
can after a little bit.
I think they're all over. These are all.
Is that them or is that one?
I can't tell.
Little though, huh?
No. Yeah, that little.
Want to try that one?
Nah, let's keep going. Now I'm getting greedy. What the-
I think there's a lot in this area.
It was on the map. There's a Scarpia tube. No,
Up ahead here, just
no,
above that
foreign.
barn.
Too small. Foreign. I don't know. Yeah, too small.
Yeah.
There you go.
Okay. Hunt is on.
There.
Oh, is it over here? Let's see. Ah, the autofocus.
No. Maybe not. Blobs.
There in front of us.
Mud balls. Aha.
Yeah.
I'm just gonna zoom up on it. I'm just gonna fly on
Yep.
this line in front of that.
Yeah, there's one. Mud ball.
Okay.
Going right.
So hard to know the scale here.
Hmm.
Can I turn the leaves on here in 2 seconds?
Oh yeah, no, don't worry.
Yeah, no
Just-
problem.
It's an illusion.
We don't know much or anything actually about the longevity of the Xenophyophores. They haven't been able to be maintained in captivity, and we don't know how fast they grow. We also don't have a good understanding of how these organisms reproduce. That said, I'm not a expert in protists, so there might be some information
Most likely
about the animals.
binary fission because
Worse, yeah,
it's
yeah, yeah.
and unlikely to be any mating.
Yeah.
There is some sexual exchange in protozoans, but very few groups do it.
Right. And for those of you who may have come on a little bit later, what we're looking at here is, believe it or not, a single-celled organism. Multinucleate, meaning it has multiple nuclei, and it is a member of the foraminifera, an organism that agglutinates. That means it sort of has a sticky outer web, and it attracts sediment particles, and it builds this intricate house. Nice grab, Chris. And there's that beauty.
Yeah. Dialed it in.
Is that quiver 3?
Yeah, A3 going in Q7. A3 larger than A2
Yeah. And it's intact.
so far.
Come on, don't jinx it.
That's a beautiful sample.
Oh, yeah.
That's exactly what you want.
Yeah. Nice, Chris.
Oh no.
Oh!
I saw it happening. I saw it happening.
Oh. All right, well, we got the technique down now.
They're really light.
Nah, that's because I was trying to like get it pulled back so we could go like that. But didn't make it.
That's gonna find out.
Yeah.
Okay, push on in.
Good news is it doesn't seem like we're
It is sitting
Xeno.
between the two.
I see it.
Whether it stays there all day, I don't know. But just so you know, have
Yeah.
a look.
We'll look.
They're both looking. You guys in unison, is
In our experience.
I don't know. Yesterday we lost a bolt out of one of our.
Oh, and it
sides,
stayed?
such a cent, and it sat on the RV all through recovery, and I found it was a special bolt because it was the first. It's an imperial bolt that I didn't have.
Special
There you
bolt.
go.
All right.
Okay. All right.
We can hope, but
Here's one right there.
all right.
All right.
Yeah. Try again.
Oh, that's a cleaner one. That's nicer.
Yeah, it is
Yes.
nicer. Yeah.
Yes. This. I have a good feeling about this one.
It's wearing, it's wearing. It's fine.
There it goes. Okay.
All right. Nice, Chris.
So I'm just leaving it on the table. Just let it lie now.
Yeah.
How are we calling this now? Did you already put an A3 in the? Okay.
This
so
is A3.
Okay. Good. Come on, there you go. Yeah. All right. Woo. Okay, that one was intact as well.
Do you want us to keep it going?
Great. Let's just do one more, and then we should head to Diane's vent.
Nice fish.
There's very nice fish.
Is that one right there?
There's a little one, yeah.
Yeah, no.
No,
They were red.
Pycnogonid. That might be one there. Let's see. No.
I gotta just red these lines. I didn't get all the sediment off me.
Okay. All right.
You can see on my other camera, it's
Oh yeah. It's
Yeah. Yeah. All right, we're gonna do a little housekeeping.
just get this.
Well, it's good to know there's nice hunting out here.
Yeah.
I wonder if they would show up on Dave's photo mosaic. If we could distinguish them.
Yeah. So in this area, we're outside the photo mosaic.
Yeah. But maybe they're there, right in front of us.
They're in front of us?
Yep. Can I zoom in on it real quick?
Just at the top of the screen. Really sediment buried? I think it is. Yeah, great thing.
So he was wondering if that's another one.
Another one that's very sediment buried.
Okay.
I think that's one too.
Not sure. I'm not sure about that one.
That's a lot more impressive for the business.
Yeah.
This one's heavily colonized. Little worms.
Yeah.
Right, can we put the lasers on before we collect it? Yeah. It's probably gonna jinx it. Right?
I think you gotta zoom out just a little bit to see
Yeah.
the thing.
Yeah. Okay. Alright. There's one.
Just come up and I wonder if they're. Oh yeah, it's on the gas light there.
Yeah. Okay. There they are. Alright. Okay.
There. Sorry.
Ah, perfect. Alright.
It's stable.
Yeah.
And not much shaking it can stand.
Well, try a little bit. See if we can get some of the big blobs off. Yeah, I'm just worried about putting that giant blob of sediment on the one that's already in there.
It's gonna take a good shake. They won't disintegrate.
I hope not. They seem to be holding up okay.
I think it's disintegrating in the background now.
Well, let's see if we can coax it in if the sticky mud stays. Do you think you can try to get it in from the side to go around the dirt clod?
Maybe.
Or all right. Come on, little guy. Oh, I got a little something.
A4.
All right. A4 partial.
A4 partial. I'm not to see.
I'm not sure we can.
If we can actually differentiate.
I'm not sure we're going to be able to do any actual experiments with them, but we can save them for DNA.
Should we go for one more? On the way to prevent?
Yeah, let's try just one more, and then keep moving in the direction of Diane's been. Okay. Oof.
Dave says that we are far enough from the closest heat flow measurement that it would be worthwhile to do one here.
Okay.
Well, just the lower left.
Yep.
There's the light on the edge of the screen.
Yeah, I gotta fly to this car. We'll be in this for half an hour.
Yeah,
Just
no
gonna
problem.
fly up and try and get all the dirt off of this. What's that?
Everything's still. Is that tube still?
Yeah.
I'm not sure that is one. I don't think that is one. Let's keep looking.
Searching.
Keep searching.
That's one down ahead of them. In front of that one, round to this.
It's over to the side.
There's some
Yeah.
there. Over on the right.
Yeah, right here.
You just you don't know.
Day.
So if we settle here, there was a suggestion by Dave Kress to do a heat flow measurement. So Dave was suggesting we could do a heat flow measurement here since we're far enough away and it's out of the other direction. So we can do that as we're collecting.
Yes.
Thank you.
So there's another one there too.
A little bit there.
Mm-hmm.
What are these guys?
Maybe infrareds. Can you tell with the tubes?
Yeah. Just makes sense.
Okay, nice grab.
Yeah. Yeah, don't put a default profile on there. So. Yeah.
Okay, here comes the tricky part.
Just go to Xeno.
Okay.
Yes. There's quite a bit of diversity of Xenophyophores that have been described so far, and they come in a lot of different types of morphology. So it is true, the ones that we find on the seamounts and in other areas have a little bit more of a rigid structure. They also have different pore sizes too. There's some with smaller pores, like the ones we're seeing here, and there's others that have a more platy structure as well. Okay. Come on. Come on. Yeah. All right. Way to go, Chris.
I was totally doing the golfing thing where, they're slicing it, sort of start leaning.
All right. That's it. We'll
That's it
We'll
for
do-
all of them, or that's it for right now?
that's it for now and for a while. Yeah.
Turn on the GoPro, but you still have to do a quick default station.
Here right now.
All right.
Alright, heat flow probe, please.
So that's eight. Was that A4?
Yep. All right. 5.
A5.
Yeah. This one's cutting off, right?
Yeah, a lot of A's today.
A lot of part.
This is the biggest sample.
Yeah, definitely. The more information you can give, the better.
It's trying to get it nice, like perpendicular on there, but
All right.
It's like campsite this far.
All right. So we were able to talk to some of our shore-based scientists, and they suggested this would be a good site to do a heat flow measurement, since we are on the periphery of the highest temperature flux, and it should be amenable, nice soft sediment, and the right thermal gradient to do these kinds of measurements here without frying the probe. So I'm gonna do this, and then we'll head off to a place called Diane's Vent that was visited on leg 2, and that was discovered in 2015. We've been going back to this site. It's pretty iconic environment, so if you haven't seen it yet, you're in for a treat.
these roads. So it's kind of like, I think I got my head on that. I was like, I can't even lift the arm. What hands? No, couldn't be us. There we go.
It was amazing. Said it was amazing. You guys good? Yeah.
I went to courses.
What's that?
I went to course, the first course.
Push course.
That's what I was
Do you?
Okay. We can insert the probe then.
Yeah.
All the listeners are in.
Oh, yes.
Yeah. It's climbing.
It could be worse than a good time. Pretty warm.
hotter than where we did the math.
Yeah.
But, you know, we're in the middle of a big vent field, so I thought it was surprising.
Alright.
I can't read with the. Is that 5?
Like 40 meters. Yeah, that's that.
I'm not seeing that. It's very
It's the
soft the edge, isn't it?
on the right hand one. Or actually on here it's
No,
under,
it's
the
off the
blue, it's the
left
blue
hand side of
mark.
the screen. It's just off the screen to the left.
No,
Isn't it?
no,
No.
it's the blue
Oh, the blue dot's on. I can't
Yeah.
see because the blue dot's on.
Yeah.
There it is.
Yeah, it's under the blue dot.
Yeah. Okay.
What were we looking for?
Diane's been
Yeah, it's right, just
Yeah.
40 meters.
Yeah.
South, east of us.
Yeah, that makes sense.
Yeah, so we left the goods. All right, so once we're done with this heat flow measurement, we're going to set up to take a push core, so we have sediment context for our Xenophyophore, and we're going to hope for the best, that the sediment stays in the tube core. Just we're talking about a little strategy on maybe how to try to do that, get things to stick a little better. While we're doing these heat flow measurements, we don't want to do any other types of activities that might interfere with the probe measurement. So unfortunately, we can't multitask, but we can look at the seafloor. So we'll take a little tour around. Pycnogonids face planting or getting something out of the sediment. Is it a pycnogonid? Cookies.
It's just essentially where we started. So we'll start here and that's it. And then just pull it on out.
Maybe not. Oh, we catch something. isopod. All right. So we have confirmation from our invertebrate experts that this is an isopod that presumably is doing a little hunting for breakfast.
Okay, good.
You got it?
Thank you, man. Ready to remove?
Let's pull it out.
Yeah.
All right, that's it all the way up.
Okay.
That's the end. Okay.
We caught a Xenophore. Oh. Released.
All right, you ready to core here?
Let's take a core. We might be able to core over this little blob right here that might be a Xenophyophore.
It's funny, there's a little more current here than we've seen in the last 4 days.
Yeah. Yeah.
What were you looking at?
Okay, yeah, I saw something that looked a little like great. So maybe right over this here. I mean, we mostly want the sediment, but that might be a Xenophyophore, so might as well try. Yeah. It's number 8. All right, hold your breath. Hang on. Hang on. Might actually stay. Chris, you're a master. Nice.
Just a core to get back on track.
Great. But there might be a Xeno on top of it.
But there might be.
Yeah. Well done. Okay, we're gonna, no, just one's fine. Now we can head over to Diane's vent. Way to go. All right, now we're making our way over to Diane's vent in the Auka Vent Field. Unlike a lot of the hydrothermal features we've been looking at the past few days, Diane's vent is unusual.
The best side
It's
up here
a much smaller,
coming in from the
vigorous
northeast looking southwest. That's the low
mound
side.
that's close to the
What
seabed
I'll do
with
is I'll come in this angle, we'll just rotate around.
beautiful geyser of fluid, hydrothermal fluid released from it. And so it's a great spot to collect hopefully some pristine hydrothermal fluid samples with the gas tight and major water samplers. So we're going to do that. There's also very steep temperature gradients in the microbial mats around this site, and we're interested in collecting some push cores from this environment.
reflector from the monument on the south side.
What's that, sorry?
It just, the reflector from the monument just crossed the 20-meter mark. So
Yeah,
distinct.
should be right in front of us.
Yeah, it's just, you can almost measure the width of it on that reflector.
This is the shifted bathing.
That filming station, hip flow measurements are hot.
What?
Hip flow measurements hot.
That's the data.
That's the data.
The data is hot.
Raquel, we want a little more.
Watts.
Yeah. Okay.
How many?
Between 4 and 5 watts
Watts.
per
Watts.
square meter.
Okay.
Did the Xeno sonar work?
We never
We didn't
really
need it.
tried it out because we
You
just
didn't
saw
need it,
them.
but
They're
but
everywhere.
you didn't
I
get it.
looked
Did
over
you
it.
look and
I didn't
you
see it.
saw
We're
the
approaching the
dots?
back side of Diane's
Yeah.
vent here.
Interesting.
But it works better with the watches.
Yeah.
Start to see a little bit of the shimmering flow.
True. Those
So
just
if
look
we
like
can-
mud balls, huh?
Yeah.
Yeah.
So let's first see if we can set up on a good shot of the venting fluid from the front and try to get a really nice image of that.
Oh yeah. They look identical.
Yeah.
Yeah.
Same spot too.
No, this is over. Wow. Similar spot.
Mm-hmm.
We've never been exactly.
Yeah.
But it's in this jungle.
Yeah.
It's closer to sea bed.
It's beautiful.
Near the sea cube and the tuna field.
Yeah, they're cool.
See the anemones and
Yeah. There are anemones around here. I think we got a few in the paper.
yeah.
They're listed. Feels like it's on a bigger mound than it once was. No? It's always
I
stuck
don't
up
know.
like this, never been flatter.
But it hasn't grown a
2015?
centimeter in
Huh.
5 years.
What you think?
The only thing that's
think.
different is that this side's a little
Okay.
more chipped off.
And I think that's just
Here's
the.
the high side there, that's the low side.
Oh no, I mean the mound
No,
under
no,
it.
no. No,
It seems
because
different
there's
shape.
nothing,
Nothing.
there's nothing
Yeah.
to fall
To
back.
change.
So
Just
don't you
change.
facing kind of southwest?
Yeah.
Point.
Is it possible? Yeah, I'm trying to wonder if we can kind of
Straight on.
So weird.
straight on, maybe at
Here,
that
a little
angle a
bit.
little bit. Yeah. I'm trying to-
Or that's the mat you're thinking of, Corey? Because I was thinking
Yeah.
this mat over here doesn't look very core.
No, over in that area.
This is 2-4.
We're heading 246. Right.
Yeah.
Wild.
Yeah, the mats that we used were these.
And you had success.
Yeah.
Oh, there's blue.
blue mats, and this was by Zasgard Archaea,
Oh, cool.
Galvin's Aerophoboda from this area, and that's also where the
All right, so on that, we're gonna want to try to get some really nice images of the event. Okay.
As opposed to that. Not so good. Bring your A game, man.
That's yeah. Yeah, maybe we can look up at it. See if that works.
oh yes, that's south. The south side of Diane's been where we,
Yeah, that's
Oh yeah.
but also where the
Yes. Did we remind Greg those are here?
Yeah, I talked to him yesterday.
South side.
South
Okay.
side.
Cool.
yeah.
You could collect that.
Awesome.
Little tiny nubber. Just amazing. When we first saw this thing, I remember being so in awe.
Yep, you were the first people to see it.
I know, yeah. Yep. Amazing.
Sure would have made my life easier that day if we found this.
I remember,
I was sweating bullets trying to get those samples because we had such a short time.
Yeah. I forgot that this was also not seen in the early discovery.
Yeah.
And same with Matterhorn, right?
So we'll do
Yep.
the first, temperature first.
Are you setting up to do the water sampling now?
No.
Oh, he's talking about. Okay. Sweet. I think they're getting ready to do that. Oh. What?
I'd just say we're going to do temperature and then major and then cast heights.
Yes. Not yet, though.
We were
a couple
well,
hours.
Yeah.
Oh.
Yeah.
Unless, let me just.
Do you want to do? We could. I guess we could do it here. Are you sitting?
Yeah, they set up to
Okay.
sample.
Yeah.
Yeah, we have to bump up because I think it's too far away. Bump up on the ledge.
It's possible we could grab some of this here.
I think that one.
Yeah. With the
it's,
anemones on it.
And it's just a little bit of active. Do you also want some inactive?
Yeah, we could get a couple.
Inactive.
Mm-hmm.
So just sitting there, so it's,
Collected any rocks from there? Have you collected any rocks from there?
I have rocks here. I'd love more.
Are they calcite?
Oh yeah, it's calcite line. This is the one with the coarse Kamina and Kakamara. But because we collect on this side, if you want to go on the other side, I'd rather get rocks from the other side.
Okay. It looks like there's also some rocks there.
Yeah,
like Matt on
definitely.
them.
That looks to me like a piece of flange. They hadn't been flanged here for a long time since that collapsed.
Yeah.
So it might be interesting to grab some of that.
Careful, careful.
Who's nervous, Adam? Are you talking about this stuff over here? Or down there?
Down there.
Down there.
Yeah.
Oh, I just almost.
What? Say again?
I'm just worried about that.
I know.
can you hit them right there?
It's making me nervous.
both?
High temperature, please.
You're not gonna be able to reach it from there.
If we bring the
We're stressing
Uh,
it out.
trail in, I might be.
So,
To get any closer.
can you do the gas tights or majors?
Let's do those first.
Why don't we do those
And
first?
then-
Well,
When we come around to
I
the
want
other
to see
side.
where the turbulent mixing is with the heat temperature, so
but
I
they're
know where to
gonna-
put the nozzles.
Okay,
But
they're gonna have to move
yeah.
to do it.
Okay. Yeah.
Yeah.
So I'm being serious though, Matterhorn or Diane?
So we're getting set up to do some fluid sampling. This vent was measured at 289 degrees in 2015 and sampled again in 2017, the same temperature. We want to see if the fluid chemistry is similar to when it was sampled in 2017. We didn't sample this vent in 2015. So we're going to try and get both gas tight and major element samples out of this. And then after we fire the bottles, we'll move to where we can get the temperature probe in and do a temperature measurement. Then we'll try and collect some rocks from this area. This is called Diane's vent. You may have noticed a metal structure. That's a monument that was placed here in memorial to Diane Adams, who was a biologist trained at Woods Hole and had started her career at Rutgers before she passed away at an unfortunately early age. Monument was put out in 2017. Her colleagues at Woods Hole as a tribute to her.
Trying to look at the well collected cores and rocks around Diane's vent previously. Because I was looking at, I just wanted to confirm samples and just Dive 194, south side. Diane's vent. We've got a couple cores, and that's where the carbonate are. There. And then previous dive. Three. These are the blue and beach mats at Diane's vent. It's just for body. Just trying to place. So if these so that PC8 is wrong. The rest of these are all in the vents. Both dives are the southwest side of Diane's vent. So I just wanted to confirm.
Yeah.
Wanting to look at that one.
Yeah. 225, so that's that way. Just looking at compass to remind myself on this one is here, southwest suburb. So we're over here. Yeah, we're basically here. So the markers here.
Yeah, that's right.
We're on the northeast side, and the marker is to the left of us, left of the vent in our view, so it's over here.
Probably have to rotate it. Still be able to,
trigger and what it looks like.
Yeah.
So these gas type bottles, once they're collected, we won't analyze the fluid on board. These samples will go back to the University of Washington to Marv Lilly's lab. Marv is one of our collaborators on this project, and he'll extract the gases, and then they'll also go to tomorrow Baumberger at NOAA, who will do helium isotopes.
Can't rotate it anymore.
Yeah.
Yeah. Hold on. Yeah, I got an idea.
So that's the top of the box. That's upside down.
Yeah, spit in it. Yeah, that's not what we're fucking with. No.
That might be the marker.
That's pretty much the end of my
That's better.
rotation.
Going down, just like straight up down. It's nice to the crab in the background.
What's that?
It's the crab on the mountaintops.
Yeah.
Oh, yeah. I was gonna say,
Yeah.
like if you could come around and in,
Yeah.
that would probably get it in, but it's, like you said, you're still right on the edge of the trigger.
Yeah, I
think
can't
you could fire it
spin it anymore, so I won't be able to get down into the vent.
Are you happy with that, Steve?
I don't see any way around it, unfortunately. So, yeah. So, okay, if we're gonna do that, if we can.
I'm gonna kill some lights here for a sec just to see.
Let's not answer that thing.
Game touching
That's your thing.
Roll down. Hold it up right there.
Yeah, we can go in like that. That looks pretty good.
I don't know what that was. Scapey.
I've done what I have with the trigger now.
I think you're still good on the trigger. I think you still have a little bit more wrist.
Rotate.
Yeah. Go down a little bit more. There we go. Obviously, it's hard to see now in here, but try to see.
Down just a touch. Actually, we're nowhere near the trigger. Oh, no, we are.
Yeah.
The gas bill there.
So he's pretty close to the sidewall there, so he's
Yeah,
Can I rotate anymore?
yeah. If you can move just
Yeah, no, can I rotate the dot more?
toward that chimney well over there, a little bit more. No, that's not good there. Yeah. Okay, we can trigger.
okay. Ready for a fire? Let me know when you're stopped.
I've exited out.
Okay. I will keep fire super slow here, and we'll watch it in this one here. Ready?
Yeah, go ahead and trigger.
Brand coming up. And I think that's a fire.
Yeah,
Brand release.
yeah, fired. We don't need to do it again. That's good.
Okay. Nice. You think good place.
Alright. Now we're with that fire.
I wonder if you just
The people on
roll all this way and just
right on top of the fire.
see if we can get it up close and rotate it. Or kick it onto the field and what's that? I wonder if you could go back to the position you were in. And you're way over there, then just rotate your gas ball in.
Oh, okay, I got you.
And then you sit over this
Yeah, yeah, yeah.
and then you should be able to go across it.
Hit the hang on. I'm not gonna make it. I feel like it. People enough this to get the picture. But yeah, sure. So, wait, let me get a picture. Yeah, that's
Yeah, well, we should be able to get it from the sea level. sure fire. Yeah. Could we skip a no and go next?
Protagonist. Since we
Okay, so now we've acquired the major bottle. This one takes larger fluid samples and takes some sort of trace element clean. We haven't talked about the chemistry of these much, but on this dive we have previously, but these vents aren't making black smoke and they aren't making sulfide chimneys because they probably have the same composition as black smoke or fluids deep in the subsurface where they're interacting with hot volcanic rocks, and they probably have lots of metals. But because we have a sediment cap on any volcanic rock here, the fluids are going through sediments and they're changing their compositions, and they become much more alkaline. They lose their acidity. That probably drops all the metal out in the subsurface as metal sulfide. They dissolve some calcium carbonate if there is any in the sediment, and they crack organic matter to CO2 at high temperatures. So the vents have a very high CO2 and a little bit of methane in them. And we want to quantify that using the gas type models. But that higher pH chemistry is what means that we don't have much in the way of metal sulfides like copper and zinc and iron sulfide, and why these vents predominantly produce calcium carbonate as the main mineral that forms both the chimney linings and the mounds.
You keep coming. I can't see that. In. That's about. We probably do it, but the problem is now you're going to need kind of open the vent with your arm, which is not good. Unless you I can bring it in. Unless you pitch way up. Yeah. If you just
We're gonna
Happy there.
no. Can we rotate and put it into the middle? Yeah.
Back off a little bit. Problem is I don't think I'm on the. There, I definitely have a thing now. So, no performance.